Okay, this graph is quite tricky to analyse. If you've got several lines doing all sorts, I find the easiest thing to do is, if you've got highlighters, is to just highlight the line and it's that you want to analyse, and it makes it stand out. So in this case we want to analyse the line for chlorophyll A because that's what the question asks. But first, just in general, what we've got here is in a leaf you don't just have one type of chlorophyll, so you have two types of chlorophyll, and you have other pigments as well that make up sort of a photosystem. Ultimately it will be electrons from chlorophyll that get excited and leave and go and join the electron carriers and go down the electron transfer chain. But in order for those electrons to become excited enough light energy needs to be harvested, and that's where the other sort of accessory pigments come in. It works a bit like a Newton's cradle, the idea that the energy is passed from these and it helps to excite that pair of electrons in chlorophyll so they can leave chlorophyll and go to be accepted by electron carriers. That's a side note. Let's just look at the data. So we can see here that when the plant's exposed to different wavelengths of light, so these wavelengths over here correspond to the red end of the visible light spectrum, and these wavelengths down here correspond to the blue end of the visible light spectrum. So we can see here that chlorophyll A absorbs the most light of around, sort of a peak of around 430 nanometers. So that does actually correspond to blue. So chlorophyll A absorbs a lot of blue light. Hardly any at all, so zero between 500 and 600 nanometers, and no absorption at all. So that would mean that if you shone light of these wavelengths at a plant or at chlorophyll A, then it wouldn't be able to harness any of that light energy at all. And then you've got another peak, not as high as the peak over here, we've got another peak over here corresponding to 670 nanometers, which does correspond with red. So you have another peak, so a plant can also absorb, as well as blue, can absorb blue and red light, which then can be used to excite a pair of electrons. Okay, I couldn't fit both the graphs on the same page, but you've got that in your data booklet. So both the previous graph and this graph can be used together. Let's just look at this graph first of all. So what can this graph tell us? So first of all, this is the amount of light energy of different wavelengths from direct sunlight. So and what we can see is there's a difference, a big difference all along. So all throughout there's a big difference, a big drop in the amount of light, so the intensity of light, or the light energy, the energy in the light that passes through the leaves. So anything that lives in the shade, any plants that grow in the shade, is going to receive a lot less light intensity, a lot less light energy than from plants that receive direct sunlight. So that does mean that there's less light energy for chlorophyll to absorb. And that's one of the points for question 2. So there's less light energy for the chlorophyll to absorb, so it's less likely that pairs of electrons will get excited from chlorophyll and pass down electron carriers. So that's one reason why plants struggle to live below large trees. Another factor as well is that there's a much smaller range of wavelengths that pass through. So here we've got a large range of wavelengths that from direct sunlight, but it's only really between so about 470 and 670 nanometers, that range of wavelengths that passes through. And we know from the graph above that these wavelengths between 500 and 600 are no good to the vast majority of plants because they cannot be absorbed by chlorophyll A. So you're talking a very, very small amount of wavelengths over here that can be used by chlorophyll A to excite a pair of electrons. So all of this means that there's going to be less photosynthesis, and probably it means that there's going to be less photosynthesis and respiration. So the store of glucose, so it's basically going to be using more glucose up than the plant's producing, so more glucose up through respiration than the plant's producing in photosynthesis. And a plant cannot survive with that. Another question that links in with this graph is the idea that plants that are exposed to direct sunlight, that carbon dioxide is often the limiting factor. So how does this show this? So if a plant's exposed to direct sunlight, light's not going to be the limiting factor. So it's not in the shade, so you've got all the wavelengths of light you have got, and each of those wavelengths has got a high enough light energy as well to excite pairs of electrons. So light's not going to be the limiting factor, so that means that the light-dependent stage of photosynthesis is not going to be limiting. You do have to put it in that much detail. Also, if a plant is exposed to direct sunlight and the light does have a lot of energy, that will also bring heat with it as well. So temperature is not going to be the limiting factor either for plants that have direct sunlight. And the second stage of photosynthesis, the light independent stage, is controlled by enzymes. Every step is controlled by enzymes. So those enzyme reactions are going to be working at a fast rate, so that is not going to be limiting either. Following on from temperature, if the light independent stage is going at a really fast rate, that uses carbon dioxide for every turn. So every turn that combines with RuBP, ribulose bisphosphate, a molecule of carbon dioxide. So it's getting used at a very fast rate, so it needs to have a high demand for CO2. So it's likely that CO2, carbon dioxide concentration, will be limiting how quickly that reaction is going, not the kinetic energy of the enzymes. Okay, I've mentioned before that if you asked to describe data, I'd do a sketch graph. So you can see on a sketch graph you'd have type of pigment on the bottom, and you'd have mass of pigment in milligrams per metre squared up the side, so that's what you're measuring. If you're then going to plot bars for each of these, you'd have the means for each bar. So we've got the 2 means for chlorophyll A, 2 means for chlorophyll B, 2 means for carotenoids, and really you'd also want to put the standard deviation bars to see if they overlap or not. One of the questions says, is there a significant difference between sun and shade leaves in the amount of chlorophyll B? And so this is how to use standard deviations in this situation. So find which mean is high and which mean is low. So this is the lower mean, that's the higher mean. From the lower mean, you add the standard deviation value on. So 2.1 plus the mean gives 92.8. And from the higher mean, you minus the standard deviation. So in this case, it's 111. And we can clearly see now that that value is still lower than that value. So that means that there's no overlap. The differences are significant. But we do need to make sure we check every single time if a statistical test has been carried out. We know if you're comparing two mean values, it would be a t-test, and there's been no t-test carried out, so you cannot see the level of probability that the difference is due to chance.